Hello boys, welcome back to another session of video tutorials, and this is meant for class 12th biology students, and this is meant for 16th July 2020. We understand from our previous sessions that we were studying about a chapter called Human Health and Diseases, and in this chapter we were reflecting on immunity. What is immunity? The overall ability of an organism to fight with pathogens. Pathogens are disease causing organisms. So immunity comes into the picture whenever there is an attack from our enemies. That is, our enemies are pathogens. And the way our system, our soldiers fight these pathogens constitutes immunity. And we said there are two types of immune systems. One is the primary immune system, and the second one is acquired or secondary immune system, acquired immune system. The first one is innate, means right from birth we develop that immune system, so it's called innate immune system. And this innate immune system has got certain barriers, certain obstacles that prevent pathogens in entering into our body. Suppose a bacteria, a pathogen, disease causing bacteria, disease causing virus, disease causing fungus, disease causing helminthes, disease causing protozoan, anything wants to enter, the first obstacle is our skin. So that is a physical barrier. And then once by chance the skin allows the pathogen inside, then we have got various glands secreting certain juices, and those juices having antibodies will fight with this pathogen. So this is called physiological barrier. Like for example, in our stomach, gastric juice is secreted, and the main constituent of gastric juice is HCl, hydrochloric acid. So hydrochloric acid fights with the pathogen and kills them. That's physiological barrier. And then you got cellular barriers, wherein you got what is called natural killer cells called macrophages and leukocytes. Macrophages and leukocytes. This will be present inside the cell. So they fight with the cells and they fight with the pathogens and kill them inside the cell. And then fourth one, we have got what is called interferons. These are glycoproteins, and they attack the pathogen whenever it enters into the cell and cause infection. So the infected cell will produce naturally interferons. So in innate immunity, these are the various physical barriers that will be protecting all of us. And there is acquired defense. Throughout our life, we will be developing certain immune systems and immune mechanisms, and these based on memory. So first time it takes time, lot of time, but second time it's quick because everything is stored in our brain in the form of memory. So we say that acquired immunity is of two types: passive immunity and active immunity. Passive immunity occurs when antibodies, against bodies, foreign bodies, antibodies produced outside the body are injected into a person to counteract antigens such as snake venom. The yellowish fluid called colostrum, secreted by mother during the initial days of lactation, has abundant antibodies. Again, you must remember the specific antibody is IgA. Ig refers to immunoglobulin A. So the mother's milk contains an antibody called IgA, immunoglobulin A, and these protect the babies from diseases. And active immunity, it is the resistance induced after contact with foreign antigens like microbes. So there will be a war between antibody and antigen. So basically you must remember, antibodies are the proteins that are produced by WBC, white blood cells or lymphocytes, and antigens are the microorganisms, say bacteria, virus, all pathogens. In general, we can say pathogens. So there is always a war inside our body between antibody and antigen. So what is the structure of an antibody? This is what you are seeing is. So an antibody is basically a protein. And the protein body, as we all of know, a protein is made up of amino acids. A peptide chains will be there. Amino acid and another amino acid is linked with a peptide chain. So as you are seeing, there will be 4 peptide chains in an antibody. 2 are small, as you are seeing on the left side, and 2 are long, as you are seeing inside. So you got 2 small chains which are called light chains and 2 long chains which are heavy chains. And you know why they are called heavy and light, depending upon the chemical content that is present. So an antibody has got a structure that can be represented as H2L2. H2 stands for heavy chains 2, L2 stands for light chains 2. And different types of antibodies are produced in our body. So namely, they are called IgA, IgM, IgE, IgG and IgD. And basically you must remember that different types of Ig refers to immunoglobulins. Now let us talk about IgA. This is secondary immunoglobulin, second most abundant in our body. IgA is second most abundant after IgG, and it is found in body secretions including saliva and tears, available in colostrum, just now I said, milk, first milk secreted by mother, localized protection in external secretions, that is IgA. Next we come to IgM. IgM, macroimmunoglobulin, largest antibody. IgM is the largest antibody. Remember this. Sometimes in your UPSC, ND exam, CBSE exam, NEET exam, this will be asked. IgM. That's why we are saying macroimmunoglobulin. It is the largest antibody. I repeat, IgM is the largest antibody. It is produced early in the primitive or primary response. Most efficient antibody it is. Agglutination it causes. Complement fixation it does. So IgM is very, very responsible. And then IgG, most abundant. It is the most abundant. IgG, immunoglobulin G, is most abundant antibody found in blood, lymph and intestine. So sometimes again, competitive exams you get this. What is the antibody that is present in our blood? So IgG. And it is predominant antibody in the secondary response. It is the only antibody to cross the placenta, and it opsonizes, enhance phagocytosis and fixes complement. So it is known for causing phagocytosis. Hence IgG is the very best antibody that is produced in our blood, lymph and intestine. And then you got IgD. It functions as antigen receptor on surface of B cells. What are B cells? Bone marrow cells, I said. And present on surface of B cells and in blood and lymph. So IgD is a antigen receptor. It does very good function. And IgE, least abundant. IgE is the least abundant, found to mast and basophil cells throughout body, mediate immediate hypersensitivity, involves in allergic reactions. Again, in competitive exams, this will be there. In allergy, what is the antibody that is produced which is hypersensitive? IgE. So we have covered IgA, IgG, and IgM, IgD, and IgE. So these are the various types of immunoglobulins that are produced in our body. And first one we talked about humoral, blood mediated. Now the second one is cell mediated immunity. And in cell mediated immunity, T lymphocytes, that is thymus lymphocytes. These are big ones, macrophages and neutrophils. So T cells identify the infected cell, which express antigen on its surface and release certain protein called cytokines, which disrupt the infected cell. For transplantation of human organs like heart, eye, liver, kidney, it's very important to find suitable donor, otherwise there will be CMI. Grafts for transplantation cannot be obtained from any animal or primate or any human being rejected by the acceptor or patient sooner or later. That is why compatibility should be there. And to avoid complications, tissue matching, blood group matching, suitable donor should be there. After transplantation, patient has to take immune suppressants all his life. And this is what you call catch 11 position. You have to have your immune system suppressed because of the organ transplantation to avoid the CMI, but at the same time you have to face every day that low immunity may invite multiple pathogen attack. So you have to live with the both. Hence, you need to take care a lot. Else, body will differentiate between self and non-self. This is the problem. Body will differentiate what is mine and what is not mine, self and non-self, and graft rejection will take place. Normally, this is a big obstacle whenever there is a heart transplantation, kidney transplantation, all this. Because the CMI will identify self and non-self. The moment a donor is ready to give and donor kidney is coming inside, but our own body will reject it because it is not self, non-self. So the doctor has to prescribe medicines which will suppress the immune system for temporary time. But then still there is a lot of risk. So this is how the cell-mediated response takes place. So you got a cytotoxic T cell and you got a normal cell virus-infected host cell and you got a protein displaying viral antigen. So T cell makes contact and releases granules. Look at that. Blue ones are getting released into the infected cell. And once they release, what happens after some time? The granules induce cell to self-destruct. It's almost suicide. The granules are making, T cell granules are making the self to blast itself. So it's a sort of suicide of the cell. Virus thus cannot replicate. This is how they do. And as we said, acquired defense mechanism, acquired immunity is of two types: passive immunity and active immunity. Active immunity, as we said, it is a slow process and takes time to give its full effective response. Do not get misled by the word active. Active immunity is a slow process. Okay? Reasons of active immunity: generated in the body due to injection of mild natured microbes during immunization, or when infectious organism enters body during natural infection, induce active immunity. It may be because of the entry of pathogen inside the body. And mechanism of active immunity: microbes enter the body and identified by B and T lymphocytes as antigen, and in response to that, body produce antibodies specific to that antigen. So this is the reason why active immunity is slow. The antibodies may be there, B cells and T cells will be there, but then the specific antigen that has come in the form of pathogen inside the body has to be acted upon with specificity. So to acquire that specificity, antigen specificity, it takes time. So antibodies then act against pathogen, that is antigen, and provides immune response. So when first time it occurs, it takes time. That's why we said active immunity is a slow process. It is the resistance induced after contact with foreign antigens or microbe. What about passive? Again, don't get misled by the word passive. Passive is a quick process because it is memory based. Does not take time to show immune response. Reasons: generated in body when ready-made antibodies of pathogens are directly injected into the body. And what is the mechanism? When antibodies against specific antigen, that is pathogen, is directly injected into bloodstream, body develops immune response against pathogen in the absence of pathogen due to antibodies. Like you are seeing now, even in COVID-19, people are asking for plasma treatment. Okay? Because the person who has recovered from COVID-19 has in his or her plasma must have developed a lot of antibodies. So these antibodies, when directly injected into the next person who is suffering now with COVID-19 severe condition, the antibodies are ready-madely available. Hence, there are plenty of chances that the person who has got severity with COVID-19 can recover very fast. That's the passive immunity. Thus, when pathogen invades body, the immune response is quick. Presence of antibody in the body. Since antibodies are directly injected into the cells responsible to produce antibodies, does not involve quick immune response. Example, colostrum, yellowish fluid secreted by mother during initial period of lactation. That is just now I said, IgA antibodies provide immunity to infant and fetus, get some antibodies through placenta. That's how it goes. So this is how various types of immune systems work. And you need to remember two things: T cells and B cells. T cells are called T lymphocytes. These are triggered by antigens, produce a clone of T cells. What do? They produce killer T cells, directly attack and destroy pathogens by secreting a protein called perforin. And then helper T cells, second type, stimulate the B cells to produce antibodies. They form protein mediators called lymphokines. Third type, suppressor T cells, keep the check on entire immune system from attacking the body's own cells. And fourth one, very important one, memory T cells. These are sensitized T cells which retain memory of antigen specificity for future, and it is lifelong. Hence, memory T cells are very, very important for all of us. And what about B cells? B cells produce antibodies and therefore generate antibody mediated or humoral, I said blood immunity. They give rise to plasma cells, that is effector B cells. The antigen specific T cells stimulate specific B lymphocytes to multiply rapidly, forming clone of plasma cells. And memory B cells, some of the activated B cells do not differentiate into plasma cells but rather remain dormant as memory B cells until activated once again by renewal attack of the same antigen. And antigen presenting cells, these are called APCs, are specialized cells which include macrophages, B lymphocytes and dendritic cells. These give signals, co-stimulatory signals. Hence, we need to understand that antibodies are of varieties and they have greater role in playing our defense mechanism.